Now let's look at specification number 12. Here, we need to understand that the different proteases in the stomach and in the duodenum work best at different pH levels. For example, the pH varies along the digestive tract. The esophagus is generally neutral. The stomach, as we know, is acidic. Moving into the small intestine, the proximal part is alkaline, and so is the distal part. In the large intestine, the ascending and descending colon are both closer to neutral. Two of the main examples of proteases are pepsin and trypsin, and we'll be learning how each of them works best under different pH conditions according to where they act in the digestive system. When we talk about pepsin and trypsin, pepsin functions most effectively in the highly acidic environment of the stomach, at about pH 2. Trypsin, on the other hand, works best in the alkaline conditions of the small intestine, around pH 8. Together, their actions complement each other. Pepsin begins protein digestion in the stomach, breaking proteins into smaller peptides. Then, once these partially digested proteins enter the small intestine, trypsin, released by the pancreas, takes over, breaking them down further into smaller peptides and amino acids. Now, looking at enzyme action, pepsin and trypsin work together to make protein digestion as efficient as possible. Their combined activity ensures that by the time the food reaches the ileum, which is the final part of the small intestine, proteins have already been broken down into much smaller peptides and amino acids. This step is vital, because these smaller molecules can be easily absorbed through the lining of the intestine into the bloodstream. From there, they're transported all around the body, where they play essential roles in growth, tissue repair, and even energy production.